Hi, I'm Tetsuya Sakurai, Director of Center for Artificial Intelligence Research and Professor of Department of Computer Science. And today, I'd like to give a short lecture about machine learning. First of all, I'd like to introduce our research center, Center for Artificial Intelligence Research. This is the website of our research center. If you have interest, please visit. This center was founded in 2017, about 3 years ago. And now we have 62 faculty members. And this center consists of two divisions. One is fundamental research about AI, such as mathematical algorithms, machine learning, big data, cybersecurity, and so on. And another division is applied research, such as healthcare, wellness, sports, medical science, and so on. And in our center, researchers from both these two divisions make some joint projects. Now, I'd like to introduce machine learning and data. Data science is all about answering questions with data. For example, what is the underlying structure of data? Does it form distinct groups? What can we learn about the future from the past data? What features of data are important? Which data samples are unusual, or which data samples are representative, and so on. And what happens if we try to classify images by machine? How can machine understand all characteristics of input data? One way is to create some rules. For example, like this figure, making a table to explain some characteristics. For a cat, fur, whiskers, horns, eyes, and so on. And how about lions, or how about a dog? So it's not easy to explain all about characteristics for input data, and sometimes it's impossible. So this is why we consider machine learning. In machine learning, we feed to the machine input-output pair of data and let it discover rules automatically. For example, if we have lots of images with labels, cat, bird, automobile, and so on, so a learning algorithm can learn the relationship between input and output. That we call model. And model is discovered through machine learning from data. Model can be applied to new data input to produce outputs. There are many machine learning algorithms with various approaches to learn models from data. And there are two kinds of machine learning: supervised learning and unsupervised learning, which I will explain in next slide. In supervised learning, we are given the training data and class labels. For example, image of cats and some other animals, they have labels. A training algorithm learns to map the input to the correct output from given examples. After training, the model can predict the class labels of new test data. Some examples of supervised training algorithms include support decision trees, neural networks, and so on. On the other hand, in unsupervised learning, we are given only input data without labels. And algorithms are applied to find patterns and relationships among the input data. By training on a large number of images without labels, a computer can group and summarize the information based on image characteristics. For example, size, color, shape, and so on. And also there are many algorithms for unsupervised learning. In biological science, data is often high dimension, such as human genome data, single-cell RNA sequence, metabolic data, and so on. And sometimes it can reach more than 10 million features. And in high-dimensional data, it is much harder to find meaningful structures. Special techniques of dimensionality reduction are used. And here is an example of how dimensional reduction can be used to extract genome type from gene variant data. And like this figure, from genome data, we can extract some groups from SNP data. And here is the research that was done in our lab in collaboration with the Chunshan Medical University in Taiwan. The goal was to identify risk factors for secondary cancer. Several factors may increase the chance of developing a secondary cancer. We divide the samples into some groups and train a supervised model in each group separately. As a result, we are able to identify some groups with different risk factors. Another example is gene signature identification for non-small cell lung cancer. Input is the dataset from the cancer genome atlas, and we are able to identify distinct gene signatures. And this is another example of unsupervised dimensional reduction for human genotype data. When the data is mapped into two-dimensional space, gene variant data forms clusters according to geographic regions, Asia, Europe, America, and so on. And this is an example of single-cell RNA sequence analysis. I will not go to details. And this is also an example of single RNA sequence analysis. And in this research, we are able to identify malignant cells with the subgraph detection. Okay, I'd like to finish my lecture by mentioning some new areas and research opportunities that machine learning opens biomedical science. Some of the areas currently emerging are precision medicine, single-cell RNA sequencing, and so on. If you have interest in machine learning application for your research, come to talk to us. That's all. Thank you.